Hi Year 9, it's Miss Rasmussen here with your second lesson for week 1. Just going to start off with a quick recap for the facts that we looked at in the last lesson. So 5 questions. I'd like you to write down the answers. Either write the questions or put them down as a full sentence. It would just make more sense for your notes. Pause the video, have a go, and then come back and check the answers. So for checking the answers, your first question is what extra 3 parts do plant cells have? Order of these don't matter, but it is vacuole, cell wall and chloroplasts. Question 2, which organelle contains the genetic material? That is the nucleus. Where are proteins made? So proteins are made in the ribosomes. Which type of cell are bacteria? Bacteria are prokaryotic or prokaryotes. It's just whether you're talking about one or many. The difference between a cell wall and the cell membrane, you could talk about the position, but what I've put down is the function. So the cell wall supports the cell membrane controls what enters and exits the cell. So anything you're not sure of, anything you've not managed to do, please make sure you do go back over week 1, lesson 1, and make sure you learn those facts from your fact book. So this lesson we're looking at microscopes, and the facts that we need to learn from this one is that there are 2 types of microscope, light and electron microscope. The light microscopes are small and portable, but they have poor resolution and low magnification. The electron microscope have good resolution and a high magnification, and then image size is equal to actual size times magnification, and they're all in your fact book for you as well. So when we're talking about microscopes, what microscopes enable us to do is enlarge images so that we can see the image in a lot more detail. So we've got here the butterfly, and you can see that's what we would see with the human eye. If you can increase the magnification by 50 times, and what this means is that this image is 50 times bigger than this one, it's magnified. We can get even more detail if we magnify it by 1,000 times, again by 5,000, and then this one, this final one, is what you would see under an electron microscope. So we can really see a lot more detail on things there. Now this increase in magnification has enabled us to see images in more detail, and it's given us a lot more understanding of the cell organelles. So here is some animal cells under a light microscope, and here is one under an electron microscope. So it's given us a greater understanding of all of the things that are actually inside the cells, which is why they're so important. Now, a couple of words you absolutely need to understand is magnification and resolution. So magnification is how many times bigger the image is. So we can see here we've got our dog, and we are magnifying this image of the nose. So we are making this image bigger. Resolution is the amount you can distinguish between 2 points. So over on this side, on the left, this is a high resolution. You can really clearly see the detail as you magnify. You can see a lot of detail between 2 points. Whereas on the right hand side, you can see although we're making the image bigger, it's not getting very clear. So that's got a low or a poor resolution. So resolution is just about the amount of detail we can see. So 2 types of microscope. We have got a light microscope, which is this one here. That's what you'd be used to seeing in your lab at school. And then an electron microscope. These are huge. So that is the sort of detail you could get on a light microscope. But if we could put it under an electron one, we could get up to this level of detail. So the resolution and the magnification on an electron microscope is much, much better, much clearer. So what we've got here is a table. It's just looking at magnification, resolution, the size, and sort of the cost and how we can prepare the materials. So light microscope can magnify up to 1,500 times. An electron can go up to 500,000. So quite a big difference there. Resolution on a light microscope is 250 nanometers. So what that means is that points that are closer than 250 nanometers, we wouldn't be able to see the detail. In comparison, on an electron microscope, it's 0.25 nanometers, so a massive difference there. In terms of the size of the actual microscope itself, the light microscope is small and portable. The electron microscope is large and we say static, it's stationary, it can't move anywhere. In terms of the costs, light microscope, it's cheap, it's easy, you just need a glass slide and that's it. The electron microscope, it's difficult and it is expensive. So the electron microscope has got quite a few disadvantages in terms of the cost. So what I'd like you to do is pause the video, copy out this table and show you the advantages in one colour and the disadvantages in another, and then pop a key underneath. Colours don't matter, it's whatever you've got to hand. Okay, so hopefully you have paused and coloured in your advantages and disadvantages, so you can check your answers here. For the light microscope, the advantages are the size, so it's small, portable, cheap and easy. For the electron, it's the magnification and resolution. Disadvantages of an electron microscope is that it is large and static and the preparation is difficult and expensive. Now I've put this one here, the magnification and resolution for a light microscope, in yellow. It's not necessarily a disadvantage that we can't get the detail. It depends on what we want to do with the images and how much magnification we need. So what we tend to just say is that the light microscope have a low magnification and a poor resolution. Okay, so next thing we need to have a look at is calculating magnification, and the thing to remember for this is I am. If you can remember I am, you can work out all of the different formulas. So I is image size, A is actual size, and M is magnification. So the image size is equal to actual size times magnification. So if you were to rearrange it, you can cover the one that you need to know. So if you wanted to know what actual size is, you cover actual size, and that is equal to image size divided by magnification. And I've just written them there up on the side. So if you'd like to pause and just jot those down, it's definitely worth having those for the rearrangements. So calculating magnification, I am. So here's an example, rearranged it for magnification. So magnification is the image size, the I, over the actual or the size of the real object. So the image of a cell in a book is 4.8 centimetres in length. However, the real cell is only 120 micrometers. Calculate the magnification of the cell. So the image, first thing we'd need to do is convert the units. Don't worry too much about how we've done this. We'll look at this in detail when we're back at the college. But what I've done is just changed it into the same unit as for the real cell. The real object then is 120 micrometers. We don't know magnification, so we need to work out the size of the image divided by the size of the real object. So we pop our numbers in and we get 48,000 divided by 120, which gives us a magnification of 400 times. Make sure, please, when you're writing magnification, you put that times down. That just says that the image is 400 times bigger than the original one. So what I'd like you to do is to have a go at your own. So if you can pause the video, copy out the table and work out the missing numbers. There is no conversion in units here. So for example, the first one, you need to work out magnification. You've got the image size here and you've got the actual size here. So you would do 10 image size divided by 0.002 actual size, and that would give you the magnification. So when you're done, if you can come back and check your answers. So welcome back, guys. Hopefully you've paused, had a go at that. Just check your answers. So on the first one, the magnification is 5,000 times bigger. On number 2, the image size is 20 millimeters. Number 3, the magnification is times 1,000. On number 4, the image size is 2.5 millimeters. On number 5, the magnification is times 100, and then on the last one the actual size is 0.5 millimeters. So please have a go at these and learn that I am image size equals actual size times magnification. So here's an exam question which I'd like you to have a look at. It's a typical one taken from actual exam papers. The average diameter of a real red blood cell is 0.008 millimeters. On the photograph, the diameter of the red blood cell is 100 millimeters. Use the formula to calculate the magnification of the photograph. The diameter on the photograph is equal to real diameter times magnification. So this is going to be our image, this is going to be our actual size, and it wants you to work out magnification. So I'd like you to pause the video, have a go, and then come back and look at a worked example. So have a go at pause and have a go at it. So welcome back. Hopefully you've paused, had a go. Just going to show you how I would approach this question and see how our approaches are similar or different. So first thing that you need to do is get the numbers that you already know. So we know that the diameter on the photograph is 100 millimeters. The real diameter is 0.08 millimeters, and we don't know the magnification. Really good practice to move these numbers down from the question because you will get some marks for doing this. Next thing we need to do is rearrange that formula. So use the triangle. We've got the I am, and we need to work out this. So magnification is going to be the image size divided by the actual size. We can then pop our numbers in using the arrow method. So if we literally just draw an arrow, so we know this one is going here, this one is going here, and put them in again. And that would actually give us 1 mark because we're showing that we can rearrange it. We know what the numbers are. So even if it went wrong in the calculator, you could still get your marks. And then put it all in, and it gives you times 12,500 magnification. It's worth putting it on there just because that makes it really clear that that is your answer. So just to recap, the facts that we would like you to learn from this lesson is that there are 2 types of microscope: light microscope and electron microscope. Light microscopes are small and portable, but they have a poor resolution and a low magnification. The electron microscopes have a good resolution and a high magnification. And then image size is equal to actual size times magnification. So make sure that you're learning those facts from the fact book. Any problems, come and contact any of the science teachers and we'll be able to help you out. And I will see you on your lesson for next week. Thank you very much.